that are very major players in proliferation. So we said, aha, this is interesting because this mirror apparently controls the amount of these genes, and when the mirror is suppressed, these genes should go up. And indeed, that's what was seen. And one of the experiments that they did several experiments, I'll just quickly mention one. You take a mouse which has a cancer, and you treat it, you inject to it or oculate it with this particular microRNA. And then you watch the growth of the tumor as a function of time. And what they observe, this is a control group. These are the mice who did not get this particular microRNA. And these are the mice who did. And you see that the tumor that in the group where, okay, I should try not to confuse this. So in the group where the mirror was injected, the tumor didn't grow. And in the control group that was not injected with this microRNA, the tumor grew. So by putting this microRNA in, you suppress its targets, which are involved in proliferation, and therefore, if you suppress those targets, the tumor will not proliferate. So this is this is the end of a series of experiments which actually shows in a living animal, in vivo, the effect of this particular microRNA. Yeah? MicroRNAs are extremely stable. One of the uses of microRNAs that people think of is to use them as for biomarkers, because even in the blood, they are there and they stay there and they don't degrade. So if you inject it, it stays for quite some time. But a good question, because many times genes, you have to incorporate it in the host cell's DNA to have it constitutively expressed in order to make sure that it's there. Whereas here, this inoculation sufficed to cause this event. I'm sorry? I will answer that afterwards, okay? I mean, I think there is a reference to the paper somewhere, it came out in EMBO Medicine last year, I think, and it's easy to find. But I'll give it to you at the end of the talk. Okay, so this is my actual talk for today on outcome prediction in breast cancer. So let's see how far I can get in this. So this is the outline of what I plan to do. I'll describe to you what is early discovery breast cancer, and the hype part or hope part is to use gene expression in order to prognosticate. I'll give you a very critical review of the way this was done, and part of it I will do today, and part of it I'll do tomorrow in this more general talk. And then I'll go try, hopefully get to this. If I don't, this will be a loss for you, because this is the current work that we are doing and I'm very excited about. So let me start. So the challenge is obvious. It's personalized prognostic predictive medicine. And the idea is to take a sample from a tumor, measure there whatever you can measure, and use all those numbers in order to prognosticate. Prognosticating means take those numbers and tell me, is this tumor aggressive or not? If I leave this patient untreated, will she, because it's breast cancer, will she live? Of course, you take the tumor out surgically and you don't just let it sit there. But after that, if you don't do anything else, will this woman live for, I don't know, 10 years, whatever the follow-up that you can, without any metastasis or without recurrence? This is the question you want to answer. Yes or no? Now, there is another question which is very relevant and more complicated, is response to therapy. There are different therapies you can try, and you don't want to do it blindly, which is the way it's done. And before you give a particular therapy, you want to know, have an idea whether this therapy is going to be effective for this person or not. The key is the word personalized. Okay. So breast cancer. So this is a disease which kills in the US about 25 out of 100,000 women per year. So it's a killer, but it's not a major killer. You saw the graphs yesterday. And the rate is going down. What is amazing about it is the incidence. About 1 in 9 women encounter breast cancer in their lifetime. 1 in 9. That's an enormous percentage. So they don't die of it. But it is like an epidemic. Okay? And what I'll focus on is what's called early discovery breast cancer, which is small tumors, less than 2 centimeters, and they have not spread to the lymph nodes, and they are of the lowest grade and stage. So this is a picture of the breast from the side, and here is the nipple, and there are ducts converging on the nipple, and the ducts on the other side of the ducts are lobules. So the milk is produced here and conducted by the ducts to the nipple, and this is the front view of the same object. Here you can also see the lymph nodes, which are axillary lymph nodes. They are here. These are the first ones to get contaminated. If there is cancer, the tumor, like every other tissue, is washed by the lymphatic fluids, which get washed to the lymph nodes, and then you will see cancer cells in the lymph nodes. That's the first place where you see them. So now, coming to define grade and stage, first of all, you can see the classification of these cancers according to whether they occur in the lobule or in the duct. They are lobular or ductal. Then you have, if the cancer is very small and has not broken out of the tissue where the site of origin, then it's called carcinoma in situ, in its site. And if it starts, then it's the lowest stage, stage 0. If it broke out of the tissue, it's stage 1 or 2, depending on the extent to which it broke out. And stage 3, it already has spread to the lymph nodes. That's stage. Grade is something else. Grade, a pathologist takes this slide with this tissue and looks. And he decides how abnormal the cells are. To what extent they are different, they deviate from the way normal cells look. And he grades that, and he gives a grade of 1, 2, or 3. 3 is this, when it already looks very different. These are the normal cells. This is the lowest grade, intermediate, high grade. So you understand now how they stage and grade breast cancer. Now, what do they do? The treatment is, the first step is resection. They remove the tumor, surgery. Then radiotherapy, they usually do that. And then if the cancer expresses a particular hormonal receptor, the estrogen receptor is overexpressed, then they give a drug which targets the estrogen receptor and blocks it. It's called tamoxifen. There is another molecule which is HER2. It's one of those receptors that tyrosine kinase receptors, you may remember. I showed you a movie which someone helped me to run. And that movie showed you the EGF receptor. One of that family of receptors is called HER2. It is overexpressed in about 15% of breast cancers. And if that happens, then they give another drug called Herceptin, which attaches to that receptor. So these are targeted therapies, and they are personalized. But there is another modality of therapy, which is the most common, systemic chemotherapy. Systemic chemotherapy doesn't target a particular molecule. It targets dividing cells. When the DNA is open, systemic chemotherapy will attack that open DNA and damage the cell. So the problem with that, it's like carpet bombing. There are cells in our body which divide, and it's part of our normal life. The cells of our intestines, the lining of our intestines, are replaced every 2 weeks. Okay? Our skin is always replaced continuously. Our hair grows. So if you give a person this kind of chemotherapy, whatever divides normally may also get attacked, and people lose their hair. Okay, so this chemotherapy it's not a good thing to have. So the question, the big question is, should you or shouldn't you give one of these early discovery breast cancer cases chemotherapy, yes or no? That's the question. And the clinicians decide on this question by using all kinds of criteria which rely on clinical indicators. And here are some of them. There is the European protocol, the American protocol, and there is a British protocol. And these are the ones that were mainly used. There is also some online software where the clinician inputs clinical indicators and an answer comes out, yes or no, or a number comes out, and according to the number he decides. So I will not go through all this, but they are, for example, all of them do the same thing basically. If it's estrogen, so they'll define a patient as low risk. Low risk means I will not give her chemotherapy. And if she is estrogen receptor positive and node negative, it hasn't spread to the lymph nodes, the tumor size is small, the grade is the lowest, and the age is above 35. Age is a very strong prognostic indicator. So if all this is answer to all this is yes, then she is low risk, and you tell her, go home, don't get chemotherapy. So these are the criteria. The question is how well they work. So this table tells you how well they work. We know that about 80% of these tumors are non-aggressive. We know because there are data banks with thousands of cases going back tens and tens of years, before all the therapies were available. And we know that about 20% are aggressive of these tumors, and these are the ones that we should try to treat. But only about 20% of them respond to standard chemotherapy. So in the optimal case, we should treat these and these only, so altogether if everything is independent, which I'm not sure about, 4% of the women should get this systemic chemotherapy. And according to the NIH and St. Gallen criteria, we end up giving chemotherapy of 80 to 80% of these women. So we are severely over-treating them, which is a big problem. And the question is, can you do better with all this hype of high throughput expression profiling and whatnot? So this is the question. And the problem is that you want to avoid, there is a bad error. The bad error is to take a person who needs chemotherapy and send her home. The probability for that you want to be less than 10%. That sets the threshold for all these criteria. And given that, you want to maximize your ability to correctly identify the good outcome patients. Are you guys still with me? Yes, because I'm a bit accelerating. Well, you could give chemotherapy to everyone, which is close to the 10%. But you have to, that error you have to keep as low as you can, but within limits, because you don't want to poison the entire population. Okay. So they look at expression profiling, and you probably saw, I only showed you some heat maps, so here is an expression profiling. What you see here is data from a colon cancer population. Each row is a gene, and there is 1,000 rows in this heat map. Each column is a patient, and the color represents the level at which this gene is expressed in that patient. I showed something like this to you before, but you have to remember, this is what people measure when they measure gene expression. This is a huge Excel file presented on a screen in terms of colors. And the aims are, let's focus on the last one, to use this expression profiling to construct a predictive classifier, okay, to try to identify good versus bad outcome. Each tumor is characterized by 5,000, 10,000 numbers, the level of expression of so many thousand genes. So each one has its own profile. So let's see whether we can somehow, using this profile, separate good outcome from bad outcome. That's the aim. Yes, yes, these samples come from real tumors. And fresh frozen and the whole thing. Okay, so what's the problem? The problem is In order to do that, to do this identification, you need a data bank. Okay? And your data bank contains all these tumors for which you measure gene expression, and you have to run a 5-year follow-up because you want to say this is good outcome, this is bad outcome. I want to learn from examples. So I need examples. And to get examples, I have to know that this expression profile corresponds to good outcome. This expression profile corresponds to bad outcome. So I have to collect samples for which I have the profile and I have the outcome. And if the question, if you define good outcome as someone who lived without recurrence and metastasis for 5 years, you have to wait 5 years. And that is a pain. So, and that is essential. Now, and that problem, once the labels are assigned, you can do some machine learning. And this was done. The first very large, at the time, effort was by a Dutch group, and they reproduced what's called a 70-gene signature. It's called the Amsterdam signature because this was a group from Amsterdam. And they wrote a paper in Nature in 2002 where they reported this. Here is a prognostic signature. There is a device which we sell. It's called MammaPrint, and it predicts with a high rate of success whether a particular early discovery tumor is good outcome or bad outcome. And then some years later, another signature popped up by a group from Rotterdam, and it's called the Rotterdam signature. It had 76 genes on the list of predictive genes. And then the question was, what's the overlap between these two signatures? And the overlap turned out to be 3. So some people were sort of raised their eyebrows and said, what the hell is going on? And then the reaction of the community was, it's okay, because they used different platforms. These used Affymetrix microarrays, and they used Rosetta. And indeed, different genes are, the same gene will be represented differently on this platform and that platform, so you don't expect the numbers to be the same. They had different populations of patients. This is also true. This is more bothersome, but because if you have a device which works only for people from Holland or Norway, then you cannot use it except there. But still, different populations of patients, and therefore not the same genes will come up, and the analysis was done slightly differently. So this is how the industry went along with the. And then several groups, us and the group in Paris, looked at this closely and wrote papers saying, no, these are not the reasons for this small overlap. So how do you make such a statement on the basis of the existing data? We had the data. Okay, so that's what I'm going to explain now. So this is the summary of this part of the talk. And I'll give it to you now because I'm not sure I'll have time to go through all the arguments, but some of it I'll present tomorrow, okay? So actually there is an inherent lack of robustness when you produce a prognostic gene list. It's not because of the platforms, it's not because of the women, it's not because of the analysis. And I'll try to convince you of that. The results that were reported in Nature and so many other journals turned out to be highly over-optimistic. This device does not produce any improvement over existing methods, classical methods, and there is zero biological insight gained. So this is strong criticism, and every one of these points I will try to establish for you. Okay, so why these failures? So this is a more philosophical slide, so I'll do it fast. There are two main reasons. One is cultural, and the second one is technical. The cultural reason for this failure of using high throughput methods for prognostication is that the field is basically dominated by two extremes. On one end, you have the machine learning guys from computer science who advocate the attitude that one of the speakers sort of jokingly mentioned here, I don't want to know the detail, I don't want to learn biology, I want to solve the problem. And this is a standard machine learning approach. Give me examples, I learn from examples. And I don't care whether the underlying problem is breast cancer of women or the choices of people buying shoes in India. Give me the vectors which you measured and the output parameter, and I will do machine learning. And I call this approach turn ignorance into a virtue. And the opposite extreme is the biologists who will tell you, how come you are trying to solve for me the problem of prognosis in breast cancer without inputting exactly the status of mutation of BRAF? Which means you need 20,000 parameters, some of which may be relevant, some of which not, in order before you start thinking about the problem. And if you say that, you are dead before you were born. So these are the cultural reasons. And there is a technical point, which was also mentioned briefly by yesterday's lecturer, Sengupta. It's called the curse of dimensionality. You have a small number of data points in a very large dimensional space, and now you try to make predictions. No go. Okay. So what does machine learning do? It takes these available samples and divides them into a training set and a test set at random. And then they take the test set and put it in a safe and lock it and give the key to a reliable person. Now you take the training set, using it by some means, which I will not elaborate on, but later I'll explain how it was done in this context. You select a subset of the genes. Those genes that you are going to use for your predictor. In the Amsterdam case, these are the 70 genes. Feature selection. Then you use these predictive genes. Now you have, let's say, 100 patients in a 70-dimensional space. You can do something. And you learn a classifier by using machine learning methods. And now you are happy with your classifier based on these 70 genes, and everything was based on the training set, on those tumors that you pulled out and said, I use them for training. Only at this point, you go to the reliable person who has the key and reveal the test set and apply your classifier on the test set and measure its performance. Now, two things can happen. The performance is good, and you write a paper, or the performance is bad, and the student doesn't get a PhD. I mean, these are the only two reasonable options. Now, what happens in the first case, the results are good, you write a paper. That's the minority of the cases, small minority. But usually, when the results are bad, you say, aha, I made something stupid. I should have done the pre-processing of the microarray differently. You say, of course, let's do that. And then you repeat the whole procedure again, and your result may improve a bit. And you say, aha, I made some other stupid thing. And then you do it again. Again, you take your test set, select a test set, put it aside, train it. This is a no-no. This is called information leak. Because unwittingly, you used the full data set in order to arrive at an improved classifier. So even though you went through the motions, you just did what you are not allowed to do. The test set you touch once, not more. Okay, so now I focus on the Amsterdam signature, and I'm going to kill it. And these are the questions that I'll try to answer. Why 70 genes? Why these 70 genes? Then so the first thing that I showed you with the overlap touches this question. But then people said, who cares? Who cares whether there is a good overlap, bad overlap? If it works, use it. Some women will be saved the agony of chemotherapy. Well, the question is how well it works. Can you assign some biological meaning to these 70 genes? And has any knowledge been gained by these genes? So I will try to answer today these two questions and tell you about pathways. So, okay, so I'll stop this part of the talk somewhere here. And in my talk tomorrow, I'll briefly go through some of this. Okay, so that's the plan. So why 70? Now, I know that we are in the Far East, and for me far, and this guy who is Moses is not very popular around here, but this is a picture of Doré, of Moses coming down from the mountain with the 10 Commandments on the tablet. He's just about going to smash the tablet. So I looked for the number 70 popping up in any of our ancient scripts and couldn't find it. So it's not there. There must be some other reason. So how were the 70 genes generated? They had 97 tumors. 19 they put aside for a test set and selected 78, the remaining ones, as their training set, and this was done at random. Random, remember, random. Random is a very important word in this game. You pulled out 78 tumors out of your pool of 97 and said, this is my training set. Then they used these 78 samples to calculate for every one of the 5,000 expressed genes on their chip, the correlation with outcome. So for every one of the 78 patients, if it's a good outcome, you give it a 1, and if it's a bad outcome, you give it a 0. So you have a vector of 78 of ones and zeros. And for every gene, you have 78 numbers, which are the expression levels of this gene on the same 78 samples. Now you dot product the two vectors, and the angle between them is the correlation of the expression of that gene with outcome. Clear. Now you take your 5,000 genes and rank them according to the value of the absolute value of the correlation. Those that are more correlated are the candidates to be predictive of outcome, and those that have zero correlation are candidates for not being predictive. So you rank your 5,000 genes, and here is the ranking from 1 to 5,000. What you do next? You say the top ranked genes will form my classifier. How many should I take? So you say, let's take 15. Construct a classifier. I will not go into the details there. And calculate the error. And this is done by a method called leave one out cross-validation. Out of the 78 patients, you leave one out. You do the whole procedure on the remaining 77 and test the outcome on the one that you left out, and you average over the 78 times that you did this. It's a way of assessing the quality of the classifier you constructed out of 15 genes. And this is your error. Then you add genes, 10 more, and the error goes down. You add more, error keeps going down, and goes down, and goes down, and goes down, and then it stops going down. Even goes up. And then you say, aha, this is my best list of 70 genes. So I'll use the top 70 out of the ranked list, not the top 700. How come 70? There are some physicists here who should immediately tell me why the number 70 came up and not 700. Okay, I don't have time to torture you, so this is the reason I am confident. They had 78 points, and they tried to fit it with so many parameters, so you cannot do better. If you make 700 parameters, then you are fitting noise. So the number 70, basically, there is nothing biological, physical, or clever in it. It was dictated by the number of samples used in the training set. So now to print them on a device and sell it for $3,000 takes courage of some sort. Why these 70? Well, I told you, they were top ranked. So, of course, if you have to take the 70, take the most correlated ones. That's the reason. But now remember, they were top ranked when you measured the correlation over the training set of 78 samples. These were drawn randomly from 97. So whenever you do something random, and this is what I teach my students, I mean, none of my master or rotation students would make this mistake. Whenever you do something random, it's okay, but do it again and again and again and check the effect of your choice. So, what we did, we pulled out 70 other patients from the same pool of 97. So of course they were not 78 different patients. There was an overlap of 60 out of them. In the two lists, we have two lists of 78. You understand? The one that they used and the one that we generated, just as they did. Now we use the new list and calculate the correlation and rank the genes according to correlation with outcome using our list of 78 patients, their patients. We have two lists of 70, and the overlap is 12. So it's not 3 as before, and I know why. Because when you pull out 78 of one pool of 97 and you do it again, the vast majority of the samples are the same. So this is not a new experiment. It's not a different population of women. Whereas in the two Amsterdam, Rotterdam, these were genuinely different tumors. Here mostly the same tumors are there. But the number is still dismally small. So this is the manner in which we established that the reason for the small overlap is not because of the different platform, because here we use the same platform to get the two lists. It's not because of different women. It was the same pool of women, of tumors. And it's not because of using different ways of choosing the gene. You use the same method of analysis. So there must be some other reason for this very small overlap, for the fact that this concept of being top ranked is not robust. Okay. I think okay. So it turns out that this is an inherently very unstable process without having to do with all these reasons that were mentioned. And let me try to explain to you the problem of ranking. Why is it unstable? So I'll start with an example, which I was called up by a Dutch journalist who wrote a piece on this problem. And this was a Dutch sport at the time, Amsterdam, Rotterdam. So he wanted me to explain to him in a layman's way what's the problem. So I thought how to do it. I explained to him. He claimed to have understood. So maybe you will also understand it without any mathematics. So imagine that there is a semi-marathon, a race. And it is held in February. So this is race number 1. There are 1,000 runners. They are not professionals. That's important. Why? Because a professional will finish a semi-marathon in 1 hour plus minus 2 minutes, 3 minutes. A non-professional fluctuates much more. His diet is not as strict as the other guy. He didn't sleep well the night before like I, and therefore fell off the podium every 5 minutes. So a non-professional fluctuates. So these are non-professional runners. And they run. And you see their arrival time. This is minutes. So the first one who wins arrives a bit more than 50 minutes. And all of them finish the race between 50 and 70 minutes, 10 minutes from an hour. So I keep putting them on, and these are all the runners, arriving from 50 to 70. And then what I do, I select a group which are the top 100. These are my best runners. Now, mind you, there are 1,000 of these guys who arrived within 120 minutes. So the average spacing between two consecutive arrivals is 1.2 seconds. That's the interval. What I do now, and this is important. So now I have a second race in March. Same runners. And they arrive, but remember, they fluctuate. Now the arrival time is fluctuating within plus minus 10 minutes of an hour of each runner. So what you will get is something like this. Those who arrived here, maybe they are a bit better than the others, but not much better. So they will be scattered all over the place. And therefore, if you look at the group of top 100 now, it will contain only a small random selection of those that happened to arrive in the top 100 in the previous race. This is the situation when the inherent noise of each runner exceeds by far the spacing between runners. These are the two things that you compare. So this is the same with genes. None of the genes in this experiment are champions. What you see here, forget this. This is a very nice colored picture. I don't have time to explain it. This is the distribution of the correlation with outcome of the genes. It's slightly shifted from 0 towards negative values. And it has a tail which is a bit fatter than this. This is the random. This is if it's a random process. And here, what you see is that the genes, the correlations of all these genes with outcome is less than 0.4, and the good ones, the best ones, are between 0.3 and 0.4. And there are about 1,000 of them. So about 1,000 runners that score within 10 to the minus 4 of each other. And when you change the training set, the fluctuation of the same genes correlation is 10 or 100 times more. Okay? So when I change the training set, go from one set of 78 to another set of 78, the same genes correlation with outcome will move by much more than the interval between him and the one that is 10 times or 100 times after him. So this is the reason for this instability. Now you can ask, what's the biology behind it? Okay, here there is a, why is correlation with outcome so noisy? Breast cancer is a very heterogeneous disease. It's not one disease, it's 200 diseases. So take a sample of 100 women, some disease will not be represented at all in your sample. And therefore, a gene which is important for that variant of breast cancer will not be represented and it will not be correlated. Now you draw another sample of tumors, and the representation of different kinds of cancer will change, and therefore everything will shift around. So the heterogeneity of the disease is the underlying reason for the large variance, large shift of the rank. And that, in turn, is the reason for the total instability of these ranked lists. Any questions about this? Probably everyone is dead tired already. Sorry? There are many ways of dividing breast cancer into classes on the basis of gene expression. There is the main classification which survived is called PAM50. It goes back to year 2000, and it's luminal A, luminal B, normal-like, HER2, and basal. And this was done by a very strong and good group of biologists at Stanford, Botstein and Brown, and this is still alive. Okay, but it's not good enough for prognosis. So there is a prognostic device based on this, which says the luminals are the least aggressive, basals and what's called triple negative are the most aggressive, and that's a basis for classification. But it should be improved. So here is a question that, so I showed you this, and I don't know if I'll have time to talk about pathway-based classification. I'll try, but we'll see. I want to show you this. In all the work that I did with biologists, I didn't use any real theoretical physics. So when people ask me, when I give talk to physicists, and at the end they invariably ask me, what of your background in physics or education in physics you used to do this, or you brought to biology? And the honest answer which I give, the only trait of physicists which I brought with me to biology is arrogance. Now, I did not mean this in a negative way. It's positive arrogance, and let me tell you what I mean. If you come to a mathematician and tell him, look, here is an important problem, breast cancer, women are getting chemotherapy, and please help me solve it. He will say, what do you want? I didn't learn about biology in kindergarten, elementary school, high school, university, and I know how to do whatever manifolds. Okay? I don't do things that I'm not an expert in. This is the mathematician's approach. The chemist, poor him, he just can't. The physicist says, okay, you know, I was in graduate school. I took a course in relativistic quantum field theory and survived it. So give me the book on cancer. I'll read it, and I'll work on the problem, see what happens. So this is what I call positive arrogance. And this is exactly what gives physicists an advantage. They come with a mathematical background, and they dare. They're not afraid to make a fool of themselves. What will their friends say? That this guy is off his rocker doing cancer biology, which they say about me in my own department, and who cares? So this was an aside. Now the main point that I wanted to make here is that usually I found myself doing stuff which is really low level in terms of its intellectual whatever. Mathematically, it's not trivial at all, and computationally, it's involved. But it's not the kind of normalization group that I did as a student. And the other extreme, to do very sophisticated stuff, call it biology, but it will be irrelevant to biology. So this is the one instance when we did something which is reasonably sophisticated theoretical physics and relevant to biology. Because we asked the following question. I just explained to you that these gene lists are unstable and not robust. And the reason is this large fluctuation of the correlation when you measure it on your training set. Now, if your training set had 1 million samples, then the value of the correlation, if you take another 1 million, would not fluctuate that much, right? So we said, let's try to find the number of samples you need in order to have typically a 50% overlap between two lists of 70 genes. Is the question well defined? So this question wouldn't be posed by a biologist. He wouldn't dream how to answer it, and it wouldn't be posed by maybe by a good statistician. But for a physicist, it's a natural question to ask and try to answer. Let me show you what you do. So what you say, let me assume that I have I generated two sets of 70, of so many patients, n patients. N is a number. And for this set of n patients, I calculated the correlation of 10,000 genes with outcome. And I identified the top 70. And now I did it again. And I calculate the overlap between these two sets of 70. I call it F. It's a number between 0 and 1. It's a fraction of overlap. It is a random variable, because the training set of n sample was pulled out at random from an enormous pool. I want to calculate the distribution of this random variable f. And I want to calculate the distribution as a function of n, of the sample size. So to do that, you have to do a calculation. And here is the calculation. So this is I will not explain what this is. There is a paper in PNAS 2006, I think, that you can look up. What you have is n genes, the number of genes, 10,000 binary variables, 0 or 1, which says whether this gene is in the top 70 or not. And for each one, you have an integral which assigns a probability from group number 1, and this is the second group. And there are 3 constraints that couple these variables. Otherwise, they are uncoupled. The first constraint says that in the first group, you have 70 in your selected. In the second group, you have 70 selected. And that the overlap is f times 70. F is the fraction, the overlap. So these constraints are delta functions, and you write them as an integral in Fourier form. And you have 10,000 variables. And you can do this using the method of steepest descent, okay, or stationary path integration, or large N expansion. Large N expansion is a respectable tool of theoretical physics. In quantum chromodynamics, they do it, and their N is 3, which is large. Here, N is 10,000. So the result that we get is pretty accurate for this integral. And what you see here, this is the resulting distribution. Only Gaussians come out of this. And f star is the most probable value of the overlap. And it depends on n, on the size of your sample. And what we did next, we took 10 studies that were published around the same time and estimated variances from the data. And for each of these studies, calculated what would be the number of samples needed if you had n, if you want f to be 50%. So this is f, the typical overlap, as a function of sample size for different cancers. The Van Twier, the Amsterdam data is this red line. And there we find that you need 2,200 samples in the training set. These are the number of samples you need in order to calculate the correlation. And only then will you have 50% overlap between 2 lists of 70 genes, ranked by correlation. Okay, so I'm proud of this. This is a cool calculation which gives a number. And to my dismay, I gave a talk about this in Santa Barbara, and Arnie Levin was there, who is one of the top molecular biologists. He discovered, one of the 3 people who discovered P53, and he's extremely smart. Extremely smart. I mean, he lives among physicists and mathematicians, and then breathes the same air, and he's really. So when I was telling this, he said, "Oh, come on." What did he say? Forget all this. I'll tell you what's the reason for this instability. And the immediate told me, breast cancer is heterogeneous. You have large variations, fluctuations, and this and that. So why bother with all this nonsense? So even he, who is as smart as they come, didn't understand that this produces a number at the end, 2,200, whereas his arguments are qualitative. So we have a problem. We have a problem to convince and to explain to this community the importance of numbers. At the end of the day, you want to know how many samples you need. The biologist's way of dealing with it is to say, okay, it didn't work for 100, so let's do 200, let's do 500, and then eventually it will work. But there is a way of coming out and saying, don't do that. Unless you have 2,000 samples, don't do this. Use some other way of going about solving your problem. It's not clear that this is the best way, but if you insist on this machine learning stupid way, make sure that you have 2,000 samples to calculate correlations and produce a predictive gene list. Okay, so do I have some time because of all the mess? Sure. Yeah. Is number specific for the data set? Yes, it's data set specific because it is governed by how much the correlation fluctuates. And in each cancer, it's different. Okay. So I'll stop here and jump to, why doesn't it jump? Yet it jumps. So this is the pathway. Now I'll try to very quickly tell you what I did so far was slinging mud on others. And now I'll try to propose something which to me seems like promise. And this was published last year, or this year, I'm not sure, in the PNAS, and it's part of the PhD student of mine. Remember, we wrote the first paper on this in 2005, which means we started working on this in 2003. 10 years later, I have a paper. We haven't solved the problem, but I think I see the light at the end of some tunnel. Okay? So, what kind of lesson can we learn from physics? And I think a main lesson is respectability of phenomenology. Phenomenology is a very respectable way of doing physics. Murray-Gell-Mann did phenomenology. Landau did phenomenology. In biology, phenomenology is a dirty word. If you want to kill a candidate in a tenure committee, you say he's doing phenomenology. So we have, I think what we can bring to biology is to show them phenomenology is respectable, it's okay, it's useful. The equation that Antonio showed, that Purcell wrote down, that's phenomenology. So we have to read the book, learn. I don't believe in ignorance, in turning ignorance into a virtue. Retain the essential aspects of the problem, build on the measurable quantities and features, and this is extremely important, use minimal models. In physics, there is such a concept. This is the minimal model which captures the essential, the symmetries. Don't make the model more complicated than it has to be. Always look for the simplest explanation. Okay? So now I'll show you a movie. I hope it works. I hope will not be offensive. And I brought it because for my Italian friends, and Michele in particular, who is not here to see it, so you tell him about it. Which demonstrates the power and the need for looking for the simplest possible explanation of a complex phenomenon. So I hope it works. It has English translation. Excuse me. Do you know what time it is? 10 past 10. Are you sure? Just like I said. Again. 7:15. It is exactly 7:15. Unbelievable. How do you know what time it is? By touching the donkey's balls. So whenever people propose complicated explanations, try to remember this movie. Okay, so now I have to get out of this somehow. Okay, so the idea is to use expression data, high throughput data, lots of numbers, but try to get as a large number of samples as you can, which is not saying much. Now, this is important. Use biological knowledge. And the way we use biological knowledge, several people mentioned these pathway center, the lists of genes that participate in a particular pathway. There is the KEGG database. I think Michele mentioned that. You also mentioned that today or yesterday, a database which has a list which says pathway, the apoptotic p53 pathway has 88 genes. Another pathway has so many genes. And there are 3 such databases that we use, not KEGG and 2 more. And the aim is to derive for each sample a number. For each tumor, derive a number which says the extent to which that pathway number 17 is deregulated in this tumor is 0.8. So you get a score for every pathway and every tumor. And the score tells you to what extent that tumor is deregulated, abnormal in this pathway. This is the aim. See if you understand the aim. And if you don't, ask. I know it's late. Okay. I'll tell you how this is derived. You do this for a few hundred pathways, and now your sample is not represented by 20,000 expression values, which will kill you. It's represented by a few hundred numbers which already have biological meaning. And for each sample, you have these few hundred numbers which tell you to what extent this tumor is deregulated in those few hundred pathways. It's a reduced representation. It's a coarse-grained variable, if you wish. And then this is the space in which you do your analysis. So the data that we use is from brain tumors, the TCGA data. It has 430 tumors and 10 normals. You need normals for this analysis. And there is another data set which has half the tumors, but more normals. Colon cancer, I'll skip this, and breast cancer. In breast cancer, there is a data set which is unbelievable. It has 2,000 tumors and 140 normal tissues. It was published in Nature last year by the Caldas group in Cambridge, UK. It's something that we wouldn't have dreamt about 5 years ago. So now, when you look at the various, for example, in KEGG, you have, and you do a histogram, how many genes are there in a pathway? That's the vertical axis. And this is the number of occurrences. So if you look, you see that most of the pathways, a vast majority of them, have 20, 25 genes in them. So if you want to understand one of these pathways, you have to look at what 20 genes are doing, not 10,000. Only a few pathways, very few, have 100 genes, so 100 or more genes associated with them. Mostly it's a few tens. So how do we calculate this pathway deregulation score? This is an example for the apoptotic pathway in KEGG, and there are 33 genes of that pathway which are expressed in our data. So each tumor, this is colon cancer, each tumor is a point in a 33-dimensional space. Because there are 33 genes. The expression level of each gene is at that dimension. So what did I do? I didn't do that. This is the only thing I touch. But everything died. Shuts things down. Will this come on or not? No, I'm explaining now how I calculate the score. So I don't know if, okay, this part is difficult to do without seeing the screen. So do what? Here, I'll try here. Oh, it's there. Okay, so it's coming. So look there or here, whichever is better for you. So look, so each point here is a tumor represented as a point, and it's in 3 dimensions because I cannot draw in 33 dimensions. Everything I'm doing it in 33 dimensions, but I'm showing you its 3-dimensional projection. It's a difference between doing the mathematics and displaying. Okay. So the tumors are projected into 3-dimensional subspace of this 33-dimensional space, and each point is a tumor. And this is colon cancer. And the colors represent normal tissue. Polyps, these are precancerous lesions. These are not malignancies. primary tumors, and metastasis to the liver and to the lung by the color. Each point is a tumor, and what I do, I take this cloud of points and pass through it a principal curve. This is a nonlinear, one-dimensional object which is closest to all these points. Now I have to be careful with the mathematicians if there are any, because I can obviously draw a curve that just goes through all the points. So I have to do some requirements of smoothing to make sure that I don't do that. And this curve represents best the variation of the points in this cloud. It's called the principal curve. It's a very respectable object, by the way. It was introduced by a rather good statistician in 1989. And there are all kinds of theorems of uniqueness and so on that I'm happy to live with this. So this captures some one-dimensional variation in the data. And now what I do, I project every one of the tumors onto the curve. So each tumor is now represented by its image on the principal curve. And the normals, which are the black ones, they all aggregate on one side, and their center of mass is here. So now what I do, I measure the distance for each point. For each tumor, I measure the distance from the center of mass of the normals along the curve. That's important. It's like measuring the distance from Bangalore to Delhi. You don't want to measure it if you travel by car. You don't measure the aerial distance. You go along the roads. So this road describes something biologically significant, and therefore I want to measure the distance along the road. And the number that comes out as this distance, that is the deregulation score of this tumor for that pathway. The tumor has an index. So I'm looking at the projection of this point. So this defines the tumor. And the pathway, because I'm doing it in the space of that particular pathway, which in this case was apoptosis. So I end up with a number for each and every one of the tumors. And it measures how different is this tumor from normal behavior in this pathway. Okay, and now I do this for several hundred pathways. There's no curse of dimensionality. I work in low dimensional space. I have much more data than dimensions. And now, this is the result for glioblastoma. And this is the only example I'll show you. I'll not show you the results for breast cancer, even though they are lovely. So I have 550 pathways, 455 samples. These are the normals. Color, again, red means highly deregulated. Blue means looks like normals. Distance from normals is low. The normals all cluster here. And then, in addition, we see that the different kinds of known kinds of glioblastoma cluster together in this representation. So what we do is biologically meaningful. This is one of those tough cases when you work hard without knowing whether at the end you did something which captures the biology or not. So the fact that there are different classes of glioblastoma, mesenchymal tumors, all of them, that's the green color bar here, all of them are together here, and they have very similar deregulation profiles. The pathways also cluster. Pathways that are sort of played similar roles have come together. All the pathways that have to do with the EGF receptor pathway are here, and you see that they are deregulated on these tumors. So what we get, we recapture the biology of glioblastoma, which is very nice, but we also pick up some new stuff. And in particular, focus on two clusters of patients, 12 and 15. These are mesenchymal tumors, proneural tumors. They are known to be of better outcome than the rest of the glioblastoma, relatively good. but there is no subdivision within the proneurals. So now we have a subdivision, and let's see whether it has some clinical implication. Okay. And to our surprise, we saw that cluster 15, this is survival as a function of time. Cluster of 15 are very good survivors, and cluster 12 are terrible survivors, worse than the rest of the glioblastoma. All of these are mesenchymal. So you ask me about existing classification of breast cancer, the situation is similar. There is a classification, and they know that basal tumors are bad and luminal A are good, but within the luminal A, there is a lot of variation, which no one has an idea about, okay, within the basals. So here, within the relatively good survivals, we found a subgroup, this, which are actually the really very good survivors, and the subgroups are very bad. We were very surprised, but we didn't have any molecular understanding of this. And some time later, let me skip this. This shows that what we do is relatively robust. We do the whole thing on another data set, and the pathways that we claim to be correlated with survival come back, half of them come back. And let me skip this as well. Okay. There is this, what you see here is in the TCGA data, gene mutations. And there is a group of patients who have a mutation in a gene called IDH1. We didn't know anything about this. This came out from our analysis. Okay? We did not input the mutation. We did the clustering, and then we looked whether there is some meaning that we can interpret in terms of mutations. And all these patients that are deregulated here and barely deregulated elsewhere have IDH1 mutations. And 2 months ago, there was a Cell paper by the TCGA group who published more detailed data, and we looked at it, and we discovered that our cluster of very good survivals, this one, 15, all those that there was one there is no IDH1 mutation, 5 for which there is an IDH1 mutation, and the rest they haven't measured. Whereas in the very bad surviving group, none had IDH1 mutation. Furthermore, these all belong to a class which is called GSIMP. It's a highly methylated set of patients. The DNA is very highly methylated. Whereas none of these is GSIMP. So we did this analysis without knowing any of this. These two clusters came up in what's called unsupervised manner on the basis of our pathway-based representation. And only a year or 6 months later came the molecular interpretation, which showed that actually what we discovered had some very profound molecular meaning in terms of these methylation profiles. Okay, so I will conclude here and skip everything else. I'll skip the explanation of how IDH1 does it. I mean, these are some of the students that I worked with, except David Givol, who was not my student. On the contrary, he's a very senior biologist who passed away a year ago, and he was my teacher who took me into all this. All these guys are scattered all over. He came back, he was a master's student, came back for a PhD, and these are the people who just did this pathway-based analysis, which I corrupted. So this is my support, and thank you very much, and I apologize for running over time for all the mess, but at least today, being more awake, I did not crash even once. So that is a positive upside of this. Okay, thank you very much.